Hello, everybody, and welcome to the EPP colloquium, and welcome to the Nowicki Lab presentation. Let's start off with the two facts that I'd like to show you on this introductory slide. And first of them is that as a geek, I am very much Team Tony Stark. And the second fact is that in the lab, we use comparative genomics. And what you see on the picture actually is a gist of the very first paper I published when I came to UTK. That paper from 2018 compared the chloroplasts of two dogwood species, Native American dogwood, which is Cornus florida or flowering dogwood, and the Asian dogwood, Cornus pusa, which is its sister species. Before I dive into the science and everything lab related, I would like to get you an overview of my scientific journey, because this hopefully will show you that science can function successfully all over the world in many different flavors. And so I graduated in Warsaw University of Life Sciences with my master's that focused on the biochemistry of nitrogen assimilation in cereals. And in that time, I also was fortunate to go to Hiroshima University for a one-year exchange program in a molecular microbiology lab, where I learned all the techniques related to that subject. That launched me on a plant biochemistry PhD in Aachen in Germany. And that project focused on lipid biochemistry and lipid biosynthesis in Arabidopsis and other plants. So that in turn landed me a job, a very short stint really, with Max Planck Institute in Potsdam, very close to Berlin, the other side of the country in Germany. And that project focused on the plant cell biochemistry and biosynthesis of pectins. The whole group was on the move across the ocean to Michigan State, and there I stayed for 3 more years doing the same research, and we analyzed mutants of Arabidopsis in pectins and mutants of tomatoes in pectins as well. So after this, I went back home for several years to my hometown actually, and there in a research institute of horticulture, I worked with vegetable plants, and we did a lot of molecular breeding regarding pathogen and environmental stresses resistance in tomatoes, in cucumbers, and in all sorts of cabbages and carrots. So pretty major crops. And finally, 5 years ago, almost to the date, I was fortunate to receive a postdoc job opportunity in UTK in Dr. Trujiano's lab. And here I worked in that capacity for 3 years as a postdoc, and then I got promoted to research assistant professor in 2019 with a focus on molecular population genetics and another focus in invasive species. And you can see that traveling around the world is actually another important aspect for the doing science. And this also brings me to the point that this job, as perhaps not many others, is a great opportunity to learn the cultures and learn the languages. In the EPP, actually, we do all sorts of things that help you bear the burden and help you release the stress related to work. Just last weekend, with the group of PIs on the left-hand side, we went for a 60-mile-long bike ride in Georgia and Alabama. And I need to stress that pets and family members and significant others and groups of friends are very highly advisable as emotional companions, perhaps much more so during the COVID stress. So we also, as a group, maybe that's something specific to our major group conglomerate, but actually with the members of other research groups as well, we hang out of the lab. So in the upper corner picture, you can see us enjoying a hockey game. Again, it was before COVID. And we also had a volleyball league that was going on for a couple of years, again, until the pandemic. The Tennessee and Knoxville area is a great opportunity for all things scientific. And here you see us hanging out at the Oak Ridge supercomputer that we toured a few years ago, which was a mind-blowing opportunity and realization of what resources we have at our disposal. But in the lab, there is fun to be had for sure, even with the daily chores or even with the less pleasant duties such as the icing the freezers. And that brings me to the lab duties and your scholarship that is your potato and meat in this department. So I would like you to introduce Shivani Sapkota, who just graduated with master's degrees and was snatched off to a great job in Athens. Shivani was my first master's student, and she worked on a series of Pyroscolariana projects. This colory pear is invasive species in North America, and many states, increasing number of states actually, declare the species as invasive, and there are policies in place and programs in place to try control it. Shivani project tackled, on the first chapter, tackled the native species and the diversity of the in the native region. And she analyzed it using 18 SSRs or microsatellites, which I'm sure you will get to know in the course of the programs. Essentially, these are molecular markers that are hypervariable and therefore very much informative for genetics, phylogeny, evolution, and all that fun stuff. And so in the first round, Shivani tried to cross-amplify 18 of those SSRs across various species of Pyrus and Malus, which is pears and apples. And you can see the cross-amplification rates in the table. On the map, you can see the geographic origins of the specimens that she analyzed. And unfortunately, not all could be shown because the GPS data were lacking. But we established that China was genetically the most closely related to a pool of the American cultivars, and Japanese and Korean samples were somewhat more related. In the second chapter, she used the same markers to analyze the diversity of the invasive populations in our region. We analyzed 3 populations from northern part of Appalachia and 3 from the southern part of Appalachia. And we concluded that they are distinct populations, genetically distinct groups, but they invaded very shortly after introduction to US. And I will be speaking about such graphs a little bit more soon. The third ongoing part of the project that is currently undergoing sequencing is sampling across many populations across the 30 eastern US states. And here I want to point out the networking opportunity, because all of these, all those 3 projects and more that are currently ongoing happened because of a Twitter contact that Dr. Hajabdish Berry shared and forwarded to me from a Clemson University collaborator of ours. Other programs that we use, such as structure here, show insights into the pathogen diversity. So here we can see the genetic groups of Pseudoporanospora humuli, which is a hop downy mildew, as genetically distinct because it has completely different color than the more complex group of Pseudoporanospora cubensis, which is a cucumber's downy mildew. And genetic programs also allow you to calculate whether each given group is multiplying sexually or clonally, with the null hypothesis being that the group is amictic and is exchanging genes freely. So if results of the analysis is not significant, we assume that this group is indeed exchanging genetic materials, but any significance of the result suggests that a group or subpopulation is multiplying mostly clonally. We can subdivide dataset for more insights and for tackling and asking more specific questions about groups. And for instance, if we do so here, we see that the historical specimens of cubensis actually showed this sexual recombination that was later lost, presumably because of the fungicide pressure on the pathogen and evolution of the resistance to them. In another project of tobacco downy mildew, Peronospora tabacina, which is also obligate pathogen, we used a program which is called do-it-yourself approximate Bayesian computation. And this program allowed us to compare it to evolutionary scenarios. On the left-hand side, you see a potential scenario which shows that the pathogen originated in Australia and through an unknown, unsampled ghost population, later split into the North American group and European populations that were sampled at the time zero, which is the current, and the whole graph goes into the coalescent. And this evolutionary scenario proved to be actually the one with the highest support, both with the direct and logistic regression. But the do-it-yourself ABC also allows you to calculate the times of splits on such evolutionary trees, allows you to calculate the population counts, and allows you to split to calculate the mutation rates for each group as well. We also tackled speciation, and for instance, we used we analyzed the common dandelion and rubber dandelion using microsatellites for insights into diversity. We also analyzed another ornamental plant, which is a tiny green and gold, to distinguish one subspecies from the whole complex using SSRs again. Finally, we used also both genomic and transcriptomic SSRs in a group of samples of specimens of Asian dogwood Cornuscusa for great diversity insights and phylogeographic insights. For instance, on the map, you can see how the current land masses are delimited with the black lines compared to the hypothetical areas that were there 20 million years ago. And so we concluded here that the species originated likely from the Sea of China, currently no longer there, currently a water body. What I want to stress for you is that making connections and networking is a major part of any scientific activities. For instance, during my times in Japan, the gentleman in the middle is Professor Katsuhiko Kondo, who is my host. And actually, he was also the host of the USDA expedition in the 1980s. That we use the samples from in the Cornuscusa project. And so I also want to brag a little bit of our recent undergrad, Logan, who graduated a few years ago and who won the Eureka University competition with the same project, and later went on to great things, including Fulbright scholarship in Mexico, and currently studies on the second year for the medical school at UT Memphis. So with that, I only have one piece of advice for you: to do your research, be it on a library and or on your computer, essentially to prepare and cut the corners and cut the fat before you jump on the activities in the lab. But also, I want you to know that we are here for you, and there is a lot of resources that are available for your use. So feel free to ask, feel free to drop on the webpage, or feel free to say hello in the corridor. And I'm really looking forward to working with you all.